Fetal gas exchange and circulation. Outline. Embryological overview. Fertilization to implantation. Maternal fetal gas exchange. Cardiovascular development. Early development. Chamber development. Maturation. Fetal circulation and fetal shunts. Transition to extrauterine life. What initially may seem like a remote concept for respiratory care practitioners, a basic understanding of embryology is essential to understanding the care of the newborn. Whether care is being provided to a premature newborn with respiratory distress syndrome as a result of insufficient or poorly functioning surfactant, or to a newborn afflicted with persistent pulmonary hypertension resulting from severe meconium staining, all respiratory care practitioners working with infants must have a breadth of knowledge of embryology and fetal development that provides them with an understanding of normal development and what to expect when something goes awry. Embryological overview. The rapidly growing embryo and fetus must develop a vascular network to circulate nutrients and provide gas exchange. The fetus depends on the mother's circulation for nutrient and gas exchange. However, the maternal and fetal vascular networks are separate systems, and no blood is shared between the two. By day 22 of gestation, the primitive fetal heart begins to beat, with myocardial pump function supporting circulation by day 27 to day 29.1. Fertilization to implantation. As the fertilized egg, or zygote, travels to the uterus, it undergoes numerous iterations of cell division but has no nutrient source, as in a bird egg. The ball of developing cells, at this point termed the blastocyst, must attach itself and implant in the uterine lining for nourishment. The outer surrounding layer of the blastocyst is the trophoblast, which combines with tissues from the endometrium to form the chorionic membrane around the blastocyst. Point 2. Inside the blastocyst, a group of cells arrange on one side in the shape of a figure 8. The central portion is the embryonic disc, which forms the three embryonic germ layers, the ectoderm and the endoderm, followed by the mesoderm. Point 3. The outer or top loop of the figure 8 envelops the embryonic structure and forms the amniotic sac, whereas the inner or bottom loop forms the yolk sac. The yolk sac soon degenerates and incorporates into the embryo, giving way for the amniotic sac to grow. Suspended in the cavity of the blastocyst, the amniotic sac then surrounds the entire embryo. The embryo attaches to the outer layer through the umbilical stalk, which later becomes the umbilical cord. Maternal fetal gas exchange. As the umbilical cord matures, finger-like projections extend into the outer lining of the chorion, or chorionic villi. Figure 2:1. Within the chorionic villi, a capillary network forms and connects to the umbilical stalk. The villi intertwine into the blood-filled lacunar cavities of the endometrium of the maternal uterus. Point 2. Oxygen, carbon dioxide, and nutrients diffuse through the vast capillary surface area of this indirect connection between mother and fetus. As fetal development continues, the region of this interface becomes limited to the discus-shaped placenta, because the amniotic sac completely fills the chorionic cavity. The umbilical cord connects the placenta to the fetus with one large vein and two smaller arteries. As the cord grows, the vessels tend to spiral. Point 4. Warden's jelly, a gelatinous substance inside the umbilical cord, helps protect the vessels and may prevent the cord from kinking. Figure 2:1. Implanted human embryo, approximately day 28, showing the relationship of the chorion, amnion, and chorionic villi. The umbilical cord and tail are difficult to differentiate in this view. Cardiovascular development. During the third week of gestation, the heart is fully formed. The heart is considered to be the first complete organ formed. By 8 weeks of gestation, the fetal heart is fully functional, complete with all chambers, valves, and major vessels. In addition, the fetal heart must accommodate the circulatory configuration required to support a fetus that is residing, growing, and maturing enclosed within a fluid-filled environment. The anatomic solutions to circulate oxygenated blood cannot be the same in the placenta-respiring fetus as it is in the air-breathing newborn. Early development. During early embryonic development, small cellular pools, referred to as angiogenic clusters or blood islands, supply nutrition to the growing embryo. These clusters coalesce to form two heart tubes lined with specialized myocardial tissue. 5 On approximately day 18, the heart tubes fold into what will become the thoracic cavity. At this point they become close enough to fuse, and they grow into a complete single-chamber tubular structure by day 21. The cardiovascular system forms primarily from the mesoderm layer, but myocardial tissue has a diverse origin related to the recruitment of myocytes from surrounding tissue types during embryogenesis. 6 By day 22, cardiac contractions are detectable and bidirectional tidal blood flow begins. 4 Chamber development. Dramatic changes begin to occur during the 4th week of gestation. The heart tubes continue to merge into three identifiable structures called the bulbous cordus, the ventricular bulge, and the atrial bulge. These structures empty into the sinus venosus, which also receives blood from three additional sources: the vitelline veins arising from the yolk sac, the common cardinal veins from the embryo, and the umbilical veins from the primitive placenta. Figure 2-2.7. These structures continue to bend, fold, and dilate by incorporating components from surrounding tissue structures as the truncus arteriosus, which connects the heart to the future arterial system, becomes recognizable. 8 Note that initially the atrial bulge is inferior to the ventricular bulge. Between days 23 and 28, a process referred to as dextrolooping occurs, whereby the ventricular bulge balloons into a C-shaped loop that pushes the atrial bulge in a superior direction. See Figure 2-2. Subsequently, the embryonic heart appears as a twisted S-shape, and the ventricular structure merges with the bulbous cordus to form a one-ventricle structure known as the bulboventricular loop, which continues to dilate. 9 Figure 2-2. Formation of the primordial heart chambers after fusion of the heart tubes at a gestational age of 3 weeks. Simultaneous with the external changes, the septum primum begins the separation of the primitive atrium, followed shortly by growth of the endocardial cushions, which will separate the atria from the ventricles. During this time, the left atrium incorporates the primordial pulmonary veins as 4 pulmonary veins empty into the primordial left atrium. The right horn of the sinus venosus grows in dominance and merges into the future right atrium from the inferior and superior vena cavae. By the end of the 4th week, the dilating ventricular spaces fold into each other and force the ventricular septal bud upward at the base of the bulboventricular loop. Figure 2-3.4. By this time, blood flow matures into a unidirectional path as the myocardium continues to strengthen by recruiting myocytes from the surrounding mesenchymal tissue. 3.6. Figure 2-3a. Sagittal view of the developing heart during week 4, showing the position of the atrium, bulbous cordus, ventricles, and endocardial cushions merging from the ventral and dorsal sides. B. Traditional view of the developing heart during weeks 4-5, showing budding interventricular septum, fused endocardial cushions, septum primum, and the left and right atria. The ventricular septum continues to fold and grow upward between the ventricles. During weeks 5 and 6, the internal and external structures continue to mature rapidly. Between the atria, the septum secundum begins to appear. By week 6, the septum secundum and a flap from the septum primum form the foramen ovale, one of the fetal shunts discussed later in this chapter. Figures 2-4 and 2-5. The atrioventricular canal continues to mature, and the endocardial cushions separate the ventricular spaces from the atrium. The muscular portion of the ventricular septum continues to grow into the ventricular space as the two ventricles dilate. Ridges also appear opposite each other in the bulbous cordus and truncus. They grow toward each other and fuse into a spiraling aorticopulmonary septum, which ultimately separates into the aorta and pulmonary arteries. 3. A fetal heart rate of about 95 beats per minute becomes discernible during this period and increases by approximately 4 beats per day until heart development is complete. 10. Figure 2 to 4 frontal view of the fetal heart between weeks 5 and 6, showing the development of the 4 chambers nearing completion. The arrow shows the one-way path through the foramen oval. Figure 2 to 5 frontal view right and side view left schematics of the foramen oval. The septum primum forms the flap, and the septum secundum remains open to form the foramen oval. The arrows show the one-way path through the foramen oval. Maturation. Continuing maturation of the internal and external structures characterizes weeks 7 and 8. The ventricles finish forcing the ventricular septum up from its base. A small intraventricular foramen remains, and blood flows between the two ventricles until the endocardial cushions fuse with the ventricular septum, see figure 2 to 4. At the end of the 7th week, tissue from remnants of the bulbous cordis and tissue from the endocardial cushions grow into the ventricular foramen, closing it as they merge with the muscular ventricular septum. The tricuspid and mitral valves form from specialized tissue surrounding the two atrioventricular openings. The aorticopulmonary septum divides the bulbous cordis and truncus into an aortic and pulmonary trunk. As these outflow tracts continue to mature, the semilunar valves form at the base of each structure. 8 Early in the 8th week, the outflow tracts and valves are completely developed. At this stage, development of the cardiac structures is complete, and blood flows through the fetal circulation pathway. The heart continues to develop, increasing proportionately more in length than width, paralleling embryonic growth. 11, 12. Fetal circulation and fetal shunts. Fetal circulation necessarily differs from circulation after the infant is born, because external respiration by the fetus does not occur within the lungs. Figure 2 to 6 illustrates fetal circulation and the 3 shunts present in the fetus that close soon after birth. The mother's lungs and liver perform most of the metabolic functions required by the same organs of the fetus. The fetal circulation pathway allows blood flow to be shunted around the fetal liver and lungs. Shunting most of the blood volume through the fetal heart, bypassing the lungs, facilitates pumping the required large quantities of fetal blood to the placenta, which is the gas, nutrient, and waste exchange interface between the maternal and fetal organ systems. 11. Figure 2 to 6a diagram of the fetal circulation showing blood containing oxygen and nourishment moving from the placenta to the fetal heart and through the 3 fetal shunts, the ductus venosus, the foramen oval, and the ductus arteriosus. Source: modified from Moore KL, editor, The Developing Human, Clinically Oriented Embryology, ed. 3. Philadelphia, 1982, W. B. Saunders. Oxygenated blood travels from the placenta to the fetus through the umbilical vein. The ductus venosus, the first fetal shunt, appears continuous with the umbilical vein, shunting approximately 30% to 50% of the oxygen-rich blood directly to the inferior vena cava, effectively bypassing the fetal liver. The amount of shunting through the ductus venosus appears to decrease with gestational age. 13 The oxygen-rich blood within the umbilical vein empties into the inferior vena cava and mixes with oxygen-depleted systemic venous blood as it flows to the right atrium. Even though some admixture occurs, the blood entering the right atrium contains the highest oxygen saturations available to the fetus. In the right atrium, most of the blood flow from the inferior vena cava crosses through a hole within the atrial septum, called the foramen oval, into the left atrium. The foramen oval, the second fetal shunt, is formed during septation of the atria, as described previously. The septum primum acts as a one-way valve over the ostium secundum, see figure 2 to 5. The remainder of the blood in the right atrium mixes with desaturated blood from the superior vena cava and drains into the right ventricle. Blood in the right ventricle contains slightly higher oxygen content than blood from the superior vena cava and is pumped into the pulmonary artery to the developing lungs. The pulmonary vascular resistance (PVR) in utero is high. Likely mechanisms include physical compression of the vessels resulting from relatively low lung volumes and hypoxic pulmonary vasoconstriction resulting from the low partial pressure of oxygen within the alveolus of the fetus. Both mechanisms help induce chemical mediators that maintain a high resistive tone in the pulmonary vascular bed. 0.14 up to 13% to 25% of the fetal blood flow presented to the right atrium reaches the lungs. 0.11, 15. Blood from the pulmonary veins empties into the left atrium and then flows into the left ventricle, out the aortic valve, and into the ascending aorta, where it supplies blood with the highest oxygen content to the head, right arm, and coronary circulation. The high PVR causes most of the blood flowing through the pulmonary artery from the right ventricle to pass through the less resistant ductus arteriosus, the third fetal shunt, directly into the aorta. This allows blood within the pulmonary artery to bypass the lungs and left heart. Because large quantities of blood are required to flow to the placenta, this permits the right ventricle and left ventricle to pump almost in parallel. The path of fetal circulation and percentage of oxygen saturation in various locations are illustrated in Figure 2-6.16. The deoxygenated blood from the upper torso returns to the right atrium via the superior vena cava. Finally, blood in the descending and abdominal aorta flows through common iliac arteries to two umbilical arteries and back to the placenta for oxygenation. 0.17 Initially 17% to 20% of the fetal cardiac output flows through the umbilical arteries, but as the fetus matures, this rises to 33%. 0.18 At any point, the placenta contains as much as half of the fetal blood volume. Because of the large vascular surface area of the placenta, impedance to blood flow is extremely low, allowing blood flow through the placenta to remain consistent and stable. Transition to extrauterine life. Clamping the umbilical vessels removes the low-pressure system of the placenta from fetal circulation. During the first breath, several factors drastically reduce the PVR and increase pulmonary blood flow. 0.19 Inflation of the lungs initiates gas exchange, which in turn dilates the pulmonary arterioles. Rising systemic arterial oxygen pressure (PAO2) also stimulates the release of endogenous pulmonary vasodilating cytokines that act locally to increase the diameter of the pulmonary arterial vasculature. 0.20 Stretching of the pulmonary parenchyma also physically expands the vasculature. Besides vasodilation, lung inflation results in the inhibition of vasoconstricting agents produced by the lung to facilitate fetal circulation. 0.19 Once the cord is clamped and the PVR decreases, pressures in the right side of the heart decrease and pressures in the left side increase. Because the foramen oval flap allows blood to flow only from right to left, it closes when the pressures in the left atrium become greater than those in the right atrium. Closing the foramen oval further facilitates the increase of blood flow to the lungs during the transitional period and is necessary to maintain normal extrauterine circulation. Because the pressure in the aorta also increases and becomes greater than the pressure in the pulmonary artery, the amount of shunting through the ductus arteriosus decreases. The functional closure of the ductus arteriosus occurs as a result of being exposed to an increase in PAO2, a decrease in PVR leading to the reduction in blood pressure within the ductal lumen, a decrease in the local production of prostaglandins, and a reduction in the number of prostaglandin receptors within the tissue of the ductus arteriosus. 0.21, 22 Normally, constriction of the ductus arteriosus starts to occur at birth, and 20% of the ductus closes within 24 hours, with 80% closed in 48 hours and 100% by 96 hours after birth. 0.23 Anatomic closure of the ductus arteriosus begins in the last trimester as endothelial tissue begins to proliferate into the lumen of the ductus, forming bulges known as intimal mounds. Initially assisted by vasoconstriction, the ductal lumen closes completely as gestational and postgestational age advances. 0.24 By 2 to 4 weeks of age, the anatomic closure is complete and blood flow normalizes to the adult pattern of circulation. 0.23 The structure that was once the ductus arteriosus is referred to as the ligamentum arteriosum in adults. EY points. The heart is the first complete organ formed. Identifying the stages of development of the heart is important in understanding congenital problems that can occur when development does not proceed as expected. The ductus venosus, foramen ovale, and ductus arteriosus are the anatomic shunts present in the fetus that allow fetal circulation. These shunts are essential to proper nutrient and gas exchange in the fetus. The three anatomic shunts and the pressure gradients induced by the circulatory systems, e.g., placental, pulmonary, and systemic, are all necessary to ensure proper directional blood flow through the fetus. This provides for gas and nutrient exchange and appropriate distribution of blood through the fetus. Transition from fetal circulation to adult circulation during the birthing process is a complex event that takes place flawlessly in nearly every birth. Recognizing the signs and symptoms of inappropriate transition is important to ensure that the right care is quickly delivered.
Placenta. Organ of respiration during fetal circulation. Exchange of oxygen, nutrients, carbon dioxide, and waste. A term occupies 1/3 of the uterine surface and weighs about 1 pound. Umbilical cord, lifeline between mother and fetus. Consists of 3 vessels surrounded by Wharton's jelly. 1 umbilical vein carries oxygenated blood to the fetus. 2 umbilical arteries remove waste and carbon dioxide. Diagram of umbilical cord showing umbilical artery, umbilical vein, and Wharton's jelly. Wharton's jelly, tough gelatinous material, prevents the cord from bending and pinching off blood flow to the fetus. Cord compression could happen during the birthing process.
Thanks for watching. For more info and videos, visit us at neonatalrespiratory.com.